you for participating, attending, and then wants to hear this talk on mutation breeding. Sir, is
Sir,
it
good
audible?
afternoon.
Good afternoon, sir. Good afternoon. Audible,
Yes,
sir?
audible, sir. It's very clear.
Okay, okay, okay. Thank you, sir. Thank you. And I'm also thankful to Director, sir, Bera, sir. Um, And then IIGR. I used to remember DGR. Now I have to say IIGR. Okay, so sir, thank you very much, sir, Bera, sir, allowing me to share my experiences on mutation breeding with all our colleagues, participants of the groundnut family. Thank you, sir. Thank you.
Yes, Director sir has joined, sir. Director sir has joined.
Muted, sir, muted.
Thank you, speaker, today's speaker, and all the participants from IIGR, and on behalf of my own self. And also thanks for accepting our invitation, and this sharing of experience and motivation of others. It is the need of the hours, because somehow we are missing something, or generation gap. I don't know. I do not know what exactly it is, but if we really want to progress and make a dent, both experience and hard work, and also resources, facilities, all these matters most. So experience and how people have contributed, paid their labor, so that signifies where today we are. So probably that insight will come, we'll listen from all the experienced and very devoted person who has made a huge contribution in their field, both classical breeding, mutation breeding, cytogenetics, genetics, and molecular breeding, even genome editing. Not only that breeding, some excellent production packages, technologies, protection technologies. So it is a very important aspect, and I am very much thankful to all the speakers for accepting our invitation in spite of their busy schedule and busy in their own field. So thank you, Dr. Anand, and very soon we'll meet in Udaipur. Thank you.
Sure, sure, sir. Sure.
Carry on.
Yes, sir. Uh, Sir, is it visible, sir? My
Yes,
slides?
visible.
Okay,
Yes,
okay,
visible and audible.
okay, sir. Thank you, sir, once again. And then I'm here to share my experiences on mutation breeding for the groundnut improvement. And then, sir, we are networking in the for the mutation breeding. Just for the mutation breeding. Particularly, we are thankful to our Indian Council of Agriculture Research. They are the main source motivation for us. In addition to that, all the state agriculture universities, they are cooperating and then helping us a lot for the development, crop improvement of our groundnut varieties. In addition to that, to reach to the farmers, the various levels, the state departments of agriculture, and the further the seed corporations, the private companies, farmers, and recently NGOs, FEOs, and all. At international level, our own mother agency like International Atomic Energy Agency, they are all helping us for the crop improvement, mutant variety development, release and notification, seed multiplication, and finally reach to the farmers. So BRC is working on these crops, oilseeds, pulses, cereals, and even in other crops like banana, sugarcane, and ornamentals. In oilseeds, of course, groundnut is the major one. I will be talking on this groundnut. This why we need the mutation breeding. Yes, Muralidharan sir has shown this thing, but still I want to repeat, particularly for our younger colleagues, that this is the one method where we can generate a newer variability for the both nuclear and cytoplasmic traits. There are very good examples even in the cytoplasmic traits. As we know, particularly our in groundnut plant habits, that is what we say, Fastigiata, vulgaris, hyposia, all those things. So they are also equally governed by the cytoplasmic factors. And then earlier days, the Ashri's group, they have done a lot of mutation studies for the cytoplasmic traits. So in such conditions, one can make use of these induced mutagenesis for creating the nuclear and cytoplasmic variability. And and also, when can we make use of this? See, already I have the best variety, well adapted variety. That is the one best thing is to start with. Okay, and then of course, if at all it is lacking in some of the traits, okay, such things can be improved through these mutagenesis. And also to break the tight linkages between the traits. As we know in groundnut, majority of the traits, particularly quality versus yields, they are negatively linked. Okay, whenever I want to increase the yield, then the quality parameters like protein, oil content will be maintained or will come down. So, or the protein versus oil also the same cases. But we have our own examples in particularly TAG 24 and the Somnath, where the some of the tight linkages have been broken by exposing to the radiation. And then, whenever we were lacking some of the sources, particularly for the disease resistances, okay, then where such genetic resources are available in the distant species, the wild species, and all. So, even if we cross, also we are sometimes are unable to transfer the genes from the wild species, or even if they have transferred, they are unable to express, maybe because of this some hiding factors may be there. So particularly the crossed ones are subjected to the radiation doses, particularly little bit of higher doses, so that we can enhance the chromosomal translocations. There are very, very good examples, success stories are available, particularly in wheat crop, where they have transferred most of the resistant genes from the rye, that is cereal. Okay, the very good textbook examples are available to transfer the chromosomal segment into the cultivated species. And then, of course, for the vegetatively propagated crops, this is the only one because where the crossing is not possible, and then whenever we wanted to have, then we can think of mutation. And then recently in banana, the wilt resistance have been developed using the in vitro mutagenesis. Means both mutation and tissue culture techniques together, they have used these techniques. So in nutshell, what I wanted to say, if at all you have things in your germplasm or already you have, then then you need not to follow this. You have your own conventional or any other method. Wherever such things are lacking, wherever I want to make some corrections, then one can think of the mutation breeding. And then, of course, mutations can be induced both radiations as well as some of the chemicals. Okay. So this is just I wanted to show you some of the audiences, those who have not seen. This is the seed irradiator. Okay. And then the seeds are kept here. Okay. And then this is the monitor. Here I will decide the dose. For example, in this case, now the just a minute. Huh. In this case, now this machine is emitting at the rate of 1.416 gamma rays per minute. If I decide 200 gray, so this is the time. So everything is set here, and then seeds are kept here. You can see here, and then this will go down. Okay, this will go down, and then the gamma source is kept here, somewhere here, so that after the this thing, it after the irradiation, this will come out. I will collect the seeds like this. Okay, this is one. The seed irradiators are available in addition to our own institute. Most of the other institutes like TNAU, GKVK, IRI, even BCKV, they are all having these instruments. Just I wanted to show you this is the gamma garden. This is the gamma feed or the gamma garden. Earlier in Mumbai, we have in our BARC also, we had a smaller type of gamma garden were there. And then in late 70s, they discontinued. But still, this is the one which is at Japan. It is maintained, usually used for tree species or some of the vegetatively propagated crops, where you can plant here your crops, and then here it is the source. Then this will in the night it will come up and then irradiate. Okay, like this, still it is operated. Just I wanted to show you elsewhere also how the things are done. This is the one, that is what I was showing you know, this center point. This is the center point is looking like this, right? So this is the one, and then this gives the radiations. Okay, this is how the plants are planted here, trees and all are planted. And then again with this also there are very good examples are there. I'm not going to tell here. And then this is another facility recently, few years back. This was created at our own institute, Rajaramana Rajaramana Center for Advanced Technology at Indore. This is where the electrons comes, and then they are accelerated so that these electrons pass through the seeds and then bring about the genetic changes. Okay, this is an additional mutation in addition to gamma rays. Similarly, this is what our few months back, my friend from Japan, they shared this slide. There are ions also there, carbon ions, iron ions. They are accelerated and then used for the irradiation also. This is now, these facilities are restricted to Japan and China at the moment. Still in India, we don't have this. Then coming back to this is the just schemes. Okay, you have a best variety irradiated, got the M1, and then grow the very large size of M2, and then select the desirable mutant, and then stabilize it genetically, and then go for the evaluation. This is one way of this thing. Or particularly in the cross-pollinated crops, where you have the parent one, parent two, like that. Still, you can think of mutation breeding for the improvement of either parent A or a parent B for some of the deficiencies, and then you can cross them again and then recreate the hybrids and then go for this thing, or the parent, sorry, mutant and that other parent, like this one can have a the schemes. Similarly, in this case, as I said, one can make use of distant species and then have the F1 and then subject it to the F1 irradiation. Particularly of our one of the variety, that is TG38, was developed through this third scheme, where the Girnar one from DGR was crossed with our parent TG26, and then F1 was irradiated and then later stabilized, developed the variety, and then got released through ICAR system through DGR the variety. So these are the different schemes. Depending on your need, one can make use of the things. Then any genotype is a response to the radiations or any EMS or any chemicals. It is a genotypic specific. So that is why just don't take it this thing from the literature. Okay, in groundnut already 200, 300 gray or whatever, simply follow it. No, each and every genotype has a specificity for the response to the radiations or the chemical. So best thing is one has to first standardize the doses for that genotype. This is one just I wanted to show here. Here, after 200 gray or so, there is a sudden dip in the growth of the seedlings. This is where one should think of, can be between 200, 300, like that. Just again, how it is done and all. So the LD50 standardization of the lethal dose of the 50% of the lethal dose, or one can think of the growth reduction that to 50%, both the character. Either one can take a germination or the seedling growth. But in my experience, the seedling growth is an ideal trait to think of the standardization of the doses. Like that, one can, using these formulas and all, one can standardize the dose. In this case, for example, based on the germination, it is 800 plus gray is the required for 50% of the lethal dose. But for the growth reduction of 50%, it is just 390. Till then, sometimes we feel this dose itself is a higher side. So sometimes we go for GR30, GR40 like that, so that it will be ideal. Because what happens, what we want is less damage, more mutations. Okay, so in that we have to balance between the viability or the survival growth and all versus the mutation induction. So accordingly, one should decide the dose based on such preliminary studies. Just you can see here, we did it for the, as I said, electron beam, and as an another mutation. You can see here GR50 for two doses. Okay, if you see here, here in TAG24, GR50 is 385, but in comes to the other genotype, it is 295. Okay, similarly, if you go to the another mutation, again, this is the difference. So there is an quite a good amount of interaction between the genotype and the mutation, like this in this example I am showing you. Okay, so anybody who can think of going for the mutation, always it is better to standardize the dose for that genotype. This is just I wanted to show you the second generation of the uh, M2 population of this thing in groundnut. And then you can see here our reactor, that chimney you can see here. This is our nuclear reactors, and then the each peg is one progeny row. So the hundreds of progenies are grown. From this, we can develop the, particularly the large M2 population to the extent of 30, 40,000 will be the ideal to identify a desirable mutation. Just I will show you the example from the same genotype, though they are academic of nature, but you can see the parent is same, but you can get the both the mutations. Like here in I am showing you the mutants from the tag 24 DOR as well as tall mutants from the same parent. Though we genetically say DORF is recessive, tall is dominant, but one can have both the type of mutations. Similarly, this is very interesting mutant. Just I wanted to share with you. This was the parent, and then from that I got one plant looking like this dwarf. Okay. Then I collected the seed and then grown in the next generation. In the same progeny, I could get all these three phenotypes, one, two, and three phenotypes. Again, when we did it further, the plants from this always bred true. The plants from this always segregated, again plants from this again bred true. This such a unusual mutants do get in the mutation study. So it is very interesting field to study in future. Similarly, you can see the another, though it is a leaf academic nature, but you see one leaf is converted into the funnel, or both the leaves converted into funnel, or only one funnel, one midrib only, or only one midrim, or both the midrim. Around in this line, you can see here, these are the funnels. Okay, you can see here, these are the funnels. Almost 20% of the leaves convert into this type of funnel phenotype. And also, till 30 days, plant is almost normal. 20 days onwards, we do start seeing such phenotypes. Okay. So so this is an mutant, okay. The late leaf spot tolerant, I should say. You can see this is the mutant from our very good established variety TG37A. This is an parental line, almost you can say 7-8 grade of LLS. Here we can say 3-4 grade of LLS. So, similarly, there are pod types. Okay, particularly so whenever you have a very good genotype, very good variety, because of the trader's trick, if they are not accepting the pods in the market for the because of the pod traits, undesirable pod traits, one can think of rectifying small such corrections. One can make it. Also, the size, pod shape, and then also the seed color. You can see here there is an increase in the seed size. There is also an increase in the seed size as well as seed color. And also this is an electron beam mutants. Okay, from tag 24 you can see the different shapes, sizes, no constrictions, short pods, constrictions, smooth pod with a beak, here only beak, less constrictions. Okay, from TG26 there is an increase in the pod size. Like that, we had developed the electron beam mutant for the first time in the country, and then some of the mutants are being evaluated at the various levels. I will show you. This is the parent TG26, and then from that we got large seed mutants. Those are being tested at different locations, and then in the Chhattisgarh ICKV, the TG89 could give 17% yield better over check, as well as the TG90 in South Gujarat to the extent of 48%. These are still being evaluated. Similarly, we could get one parental line TG66 had a folic acid content of 55%. Then we subjected these to the both gamma, that is physical, and sodium azide, that is chemical. Combination of both physical and chemical also can give sometimes better results. In this case, we could get 70% folic acid mutant. When at a molecular level, when they tested it, we could see these are the changes at the sequence level, DNA sequence level in the that AHFAD2A gene. and which could result into tryptophan to arginine and aspartic acid to the aspargine. Okay, means biochemical changes also can be brought. Using this mutant, we could develop in the background of TAG24 the other breeding lines. These are the two lines having the TAG24 background with the increased oleic acid. Similarly, with the 37A background with the increased oleic acid to the extent of 60-65. And then from TG51, we could get up to 60 to 70%. From TPG41, we could go up to 70 plus. Okay. So sometimes, though the mutant may not be desirable yield point of view, but having one unique trait, in this case high oleic acid, that can be combined with your well-established varieties. Similarly, we have also developed through crossbreeding. We have these genotypes using the source from IG, that is IGKV, ICGV 15027 having high oleic. We have now the high oleic lines in the background of TAG 24, 51, and then the Larseed TG89. Okay, with the 80 plus oleic acids. Similarly, just in this table, I wanted to show we have read in our typical genetic classes that monohybrid segregation, dihybrid segregation, 3 is to 1, 15 is to 1, like that. Even in this case, we could get some of the segregations like 5 is to 1. Sometimes we say that no, 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 some mistake is there, something. But when you advance it to the F3, F4 like that, you get some of the unusual segregations, what we say preferential segregations. Of course, 13 is to 3 is there as a suppressive gene actions, but we have heard of maternal inheritance. But in this foliaceous stipules, we have the paternal inheritance. And then even in the seed hardness, though hardness is recessive, but it has a paternal inheritance. So like that, this is the chart you can see in the flow chart. The way back in 1957, a genotype or a variety taking from UAS Dharwad from those days, the mutation breeding work started with X-ray irradiation at BARC in 1957. And then in that, so many mutants we got. Among the mutants, two direct mutants, that is TG1 and then TG3, they got released. The red square ones are released for the commercial cultivations. And then red ones are from the BRC and the blue ones are from the all our agriculture universities. Like that, I am showing you here the mutant into mutant crosses or mutant into mutant derivatives. Okay, and then another mutational event of in 1977 led to a mutant, which again in from JL24 in 84. Like that, if you come across, there's so many mutational events, and then their combinations led to almost 17 varieties from BRC, and then the another 15 varieties from the SAUs. And then just I'm happy to note some of our recent varieties had parents from the BRC like TAG 24 from Tirupati, then GG21, that is TPG 41, 35, TG37A, and then RG 638 is having another TG37A, like that. Right? So this is just to show you some of these earlier photographs of our varieties. Then just I wanted to show this, Somnath. Muralidharan sir told in our previous lecture that we have hypogaea, then the fastigiata, in hypogaea, absence of main stem flowering, and then the alternate branching, like that. This is the one having the main stem presence of main stem flowering and also having the sequential branching pattern, but having a spreading habit, which is usually observed in Virginia runner type. So sometimes we call it as a Spanish runner. Okay. So just academic in nature. Of course, it has large seed varieties and then released for Gujarat. Similarly, these are the popular varieties grown across the country like TAC 24, TG37A, then TG38, TG51. TAC 24 is the very prestigious variety, and then why is it so? Though it is having the semi-dwarf plant habit, which is good for high plant density, high planting, and then permits pegs to enter into the soil, and then the small, thick, dark green leaves, and which is helpful for enhanced radiation use efficiency and water use efficiency, determinate flowering, which can help us to have synchronized pod maturity, enhance the diversion of uh, assimilates, early maturity, and then the better partitioning. These are the things which could help to adapt in the larger area, the TAC 24. Similarly, the TG26, in addition to all the earlier traits, the best trait which is very much required nowadays is the fresh seed dormancy of 3 weeks. Okay, so because of the unseasonal rains or the extended rains, the pods used to germinate. So we can take care of by having the dormancy. So this is where some physiological studies we could do compared to our earlier parental lines like TME2 and JL24. Okay, these are having the less growth, less plant height. Okay, compared to these, but the overall crop growth rate is very high. That is having with a similar vegetative growth. You can see here it is very similar vegetative growth, but when it comes to the pod growth in these two genotypes, it is having the higher pod growth compared to this. That is also the seed growth rate followed by. This is very helpful for better partitioning. With the same filling period, you can see here effective filling period is around 60 days or so. So these are the ones which could contributing to the better yield in these two genotypes. And then we have, you can see here, the Chico is the earliest maturing genotype with 85 days or so. Then using our crossbreeding, we could develop the genotypes maturing in 90 days. Then the TAC 24 with 105 days, but improved yield. But later what we did, we could have the improved yield along with the maturity of Chico or so. Among them, one of the line we could release is the TG51. Then coming to the large seeds, we have very good large seed varieties. Of course, this is the TPG41 with 60% oleic acid and then the 3 weeks dormancy and then released to ICAR system. Then the later TG39 released for Karnataka and Rajasthan having 59% oleic acid with a maturity of 120 days and TLG45 little bit less maturity and then still it is being cultivated in comparison to 37A. This is where just I wanted to compare the seed size versus maturity. Okay, these are negatively related. The higher the seed size, the more the maturity is needed. Like we have the BAU 13 or earlier days, Asha was there, that is Ikrisat line. They were maturing with 130 days plus or so. Then our first mutant was maturing in 130 days with the large seed varieties. Later, what we did, we concentrated to bring down its maturity. We could successful to come to the extent of 120 days or so. Then the later to 115 days by maintaining the larger seed size. Of course, they are with the 60 to 80 plus or so. Among them, these three varieties got released. That is TG39, 45, and 47. That is Bhima variety. Later, we could increase the seed size to the extent of almost 140 gram HKW. Of course, still the maturity is 120 plus only. Okay. These are the ones just to show you how they are in this. Of course, they are very, very large seed. And then to have this type, we need very, very good agronomy and then very high management practices. Okay. So this is the one, the latest variety that Tag 73 was released in collaboration with PDKV Akola and also the JAU Junagadh, and then having the more three-seeded pod traits. Again, sir, this is a direct mutant of again TG38. Means what? TG38 itself is a mutant derived from the F1 cross hybrid, and then from that this is another mutant, so you can say that mutant of mutant. Okay, so similarly another variety Chhattisgarh, last year only it got released. Of course it is not a mutant, it is a cross derivative released in collaboration with IGKV Raigad, Chhattisgarh. Of course, it is doing well in both the seasons, which is very much helpful for our seed multiplication. And also, it is having 2 to 3 weeks of fresh seed dormancy, so that which can be cultivated even in the extended rainfall conditions also. These are the across the states. Like that, we have the mutant germplasm maintained some 200 plus or so. Similarly, these are the breeding lines. We have reached up to TG114, affecting most of the plant traits. Almost some 300 plus mutant and recombinant germplasm is being maintained. And then the success of our varieties and then the spread of the varieties is because of the supply of the breeder seed constantly. Last 2 3 decades or so, more than 7,000 quintals of breeder seed is being supplied almost all the states of our the country. And then particularly the National Seed Corporation and then the Gujarat state, then later, the earlier days in Odisha government has this thing. And then in the initial period, particularly the PDKV Akola has multiplied the Tag 24 in a bigger number. Because of that, the variety could spread. Just this is the one of the breeder seed plot. And then just I wanted to show that in addition to BARC, almost all the agriculture universities have multiplied the breeder seed of the Trombe varieties and then supplied to the all our seed agencies. We are very, very, very thankful to them. So because of that, the Trombe varieties could reach to the different places of the country. And then just I wanted to show you these things. Of course, these are you all have seen this, but particularly for our younger colleagues, I wanted to show. See, the groundnut is a very difficult crop. Expansion is very, very difficult, labor intensive. So these are the some examples where even through intercropping also we can push this crop. Particularly, I wanted to show the examples of Maharashtra, where in the initial period of sugarcane belt, we can push this crop for 3.5, 4 months. Particularly, early maturing variety can be pushed, so that this is how we can add the groundnut area in the existing area of the other crops. We can encroach the other crops, I should say. Okay. So this is one example I wanted to show in Maharashtra. And also in Maharashtra, of course, we can make it more economical. Groundnut, we can make it more economical by introducing the another crop like you can see here onion, so that both the crops will come, and then it is very high intensive planting. So this is how we can make it groundnut in addition to other crops more profitable also. Again, in this case, the sweet corn, and then this is also helpful, particularly in recent times. Our sclerotium is building up a lot, so that if we go for intercropping and all, such incidences can be minimized, particularly the other graminae family, means cereals. If we increase, spread can be minimized. Similarly, the lot of banana area is there. Okay, and then sir, here our one of the variety could give almost what I should say 3.5 to 4 ton per hectare in addition to initial period of banana. Okay, this is how we can make groundnut expansion as well as banana also more profitable. Similarly, moringa, though little bit shade is there, but farmers are growing. This is how they are making use of their space better. And then the initial part of our oil palm. These are these are examples from Andhra Pradesh. And then the initial planting of some of the teaks, because shadow is less from the tree species, one can expand. Of course, we have also done a lot of work on the moleculars and then identified the molecular markers, and also from the QTLs for the various characters, brucides, the phenolics, flavonoids, plant habits, seed size, rust and late leaf spot resistance. Okay, and then recently also all these things are published. You have the references here. Like that, in BRC, more than 70 varieties are released in different crops. Not only BRC, in India, more than 520 varieties are released across the species, across the crop groups. And then I wanted to tell you that the mutation breeding can be extended to the already in the existing hybrids, very popular hybrids. One can make use of improvement of their inbred lines, or we can apply these mutation breeding for improvement of the minor millets. Now the lot of area is coming up, very good price is coming up, is available. So for plant type and all, we can make use of mutation breeding. And then also for the organic farm practices where we need different plant types, one can think of using mutation breeding. And then the unexplored medicinal and aromatic crops, and then the already existing land races. We have a very good example from Chhattisgarh in rice. We have improved in collaboration with IGKV the land races of the existing rice varieties. And also we can think of improving the plant type for mechanical harvesting, for biofortification, and vegetatively propagated. And then also in addition to gamma rays, we have these ion beams, proton beams, electron beams, neutrons, or even very, very good chemicals are there where we can have the point mutations. One can expand the mutation breeding. Of course, there are limitations are there. Still, it is a random process. You cannot get a desirable mutation. One has to try a lot, and then you have to have very large populations. So then only one can think of desirable mutant. Just to give you one example, in rice herbicide tolerance, in more than 1 lakh population, only one plant was surviving. By picking that, they could identify herbicide tolerant rice mutant, that is robin. They later they transferred that trait to the Pusa basmati. Now we have the very good Pusa basmati with a herbicide tolerance. And then these desirable mutations are oftenly associated with undesirable. So for that, one should go for the back crossing, and then oftenly The mutations produce pleiotropic effects. And then, as you know, the yield and its components are quantitative in nature. To have a mutation for quantitative traits is very, very difficult, but still it is possible. We have a experience in that, and most of the mutations are recessive, whose detection is almost impossible in clonal crops, difficult in polyploid species. Our groundnut is again an allopolyploid, allotetraploid. Again, still it is a difficult. Okay. So with this, and then these all the contents are available, freely available in these books. One can read these things. Thank you very much.
Thank you, sir. Thank you. The seminar was very good, and thank you for bearing with the internet disruptions today. And, thank you, sir. We are connecting from home because
Yeah,
you don't have access to Zoom at your facilities.
yeah.
Thank you, sir. Thank you, sir. Presentation was very nice, sir. And now I open the session for question and answers. Any participant can ask question. Raise your hand and please ask question.
Good afternoon, sir.
Good afternoon. Good afternoon.
Sir, I just want to know that the bitterness in some of the varieties, they are too bitter. So whether it can be rectified with the help of mutation BD?
Yes, sir, possible. There are examples, particularly in, as I remember, in one of the pulses, no, that Kesari, Lathyrus, or neurotoxin. Okay. So there are examples. They have rectified this. Only thing is, there the screening method is important in the sense that you should have a very large population. You should have a very large population, and then you have to taste every fruit or
Yeah.
whatever your seed. Okay,
Yeah.
that is the most important, because the taste or the whatever you should make out only through your own physical testing. As I said, 20,000 plus tasting is itself is a very laborious, but possible.
Sir, one more question. We do have that SB11 variety in our Jalgaon district. It is very much popular. But
Sir.
the thing is that it is being affected by the leaf spot disease, and it is getting washed away. Means there is a very less yield, and it is very much popular because of its sweetness in taste. Whether we can have the mutation in such variety which is very pretty old.
It is not the old or new, sir. Important is what farmer wants. Is it the yield or is it the disease resistance?
Oh.
Okay. If it is the yield, okay, SB11 is very, very low yielder. So we have to give them, we have to educate them. There are very good varieties compared to SB11 yield point of view. Okay. How much the yield is suffered because of the disease resistance? Okay, so if they can take it up, one or two sprays, well and good. Or you have already a disease resistant sources are available, like GPBD4 is there, or even some of the newer varieties are there having the disease resistance. One can cross it and then go for that. Okay, but still improving SB11, that is 1940 variety, for resistance at the moment is not desirable in my personal point of view.
Okay, sir. Okay. No. Thank you, sir. Thank you.
Thank you, sir.
Veena, sir. Ashok Kumar Veena, sir.
Myself, I want to ask, sir.
Shrikant, sir. Boliye.
Sir, very good lecture, sir. Actually, your lecture are always good, sir. Thank you very much. Sir, just I want to one question about that how the dose standardized in groundnut, sir. At two doses LD50 and GR50, you have explained, sir. So how to calculate GR50 dose, sir, or LD50? That's some information that I want, sir.
Very simple, sir. First, you identify the parent what you wanted to subject to the mutation breeding. Okay. Now, you subject that to the serial doses like from 100, 200, 300 up to 1000. Okay. Have a 2 to 3 replication each with 100 seeds. Okay. Irradiate and then subject it for the germination and then look for the germination percentage. At what level more than 50% germination is affected, that is one criteria. Okay, followed by you keep the germinated seed for the seedling growth. Okay, like in our seed labs, no, we allow them to grow some 15-20 days so that your seedlings will grow, or you even still you can put it in the test tubes with the moisture so that the seedling will grow, and then you look for the seedling growth reduction. Okay, at what level the reduction happens, though the seed germinate but it may not grow. Okay,
Yes,
so
sir.
that is the beauty. So the growth affected dose you should take it, and then as I showed you in that example, you can fit into that one. That is one way of analysis is probit analysis. You subject the reduction percentage, convert them to the probit, then look for the 50%. Even sometimes, as I said, 50% is a higher side. We do go for 30% or so. Then identify those again, 2-3 doses around that you can go ahead.
Yes, sir. Sir, is it same for other? Because this one groundnut is seed propagated, but just I applied for turmeric crop, vegetatively propagated. So
Yeah,
really,
yeah, definitely, sir. Definitely, because in turmeric, only thing is, though you need to standardize the dose, but that is equally important, sir. Because what happens after subjecting, when you take it up very large scale, then when you don't get the results, then you regret. Okay, best thing is initially itself you should standardize the ideal dose because it is very, very genotypic specific, very genotypic specific. So only thing is initially you have to spend a lot of time for the standardization. Once you standardize, fantastic.
yes, sir. Thank you very much, sir.
Meena, sir. Ashok Kumar, Meena. Unmute करें sir, unmute.
Sir, my one single question is, can we use physical and chemical mutagenesis in combination? And also, what will mutagenesis we do use freshly harvested seeds, or after the moisture level is down to 8 to 10%, does the moisture level affect the mutagenesis? Are there any epigenetic changes? How do we identify them? These are some my doubts.
Sir, first is one can use a combination of mutagens, both physical and chemical. There are very, very good examples are there. And also we have our own, as I showed you, one example of folic acid. Okay, that is a very good attempt. But equally, you can have physical, chemical, and together. Okay, so that you can appreciate both the interactions. That is one aspect. And then second is freshly harvested. Yes, you can take the seed. Only thing is, as you know, higher the seed moisture content, more the damage will be. Okay. So because what happens, the water and the radiation, they interact together and then releases the reactive oxygen species. They in turn damage our DNA strand. So because of that, there will be lot of damages. Okay. Of course, those damages we need, but how much those damage will translate into desirable mutation is a question mark. Because of that, better to have a dry seeds around 10-12% of seed moisture will be ideal.
Thank you, sir.
Good evening, sir.
Hello.
Yeah, good evening, sir. Narendra, sir. Bole.
First of all, sir, congratulations for nice presentations and very informative. That
Yes.
one mutation breeding in groundnut. My 2 or 3 questions are there. First of all, sir, all the bark varieties are developed through radiation mutagens.
Hmm.
All, beside the radiation mutagens, is chemical mutagens are working in groundnut or not, sir?
Yeah, no, sir. Any mutagen for that point of view need not, any mutagen for that point of view are okay for inducing the mutation. Is it a physical or is it a chemical? Okay, both are acceptable. And then see what mutation does, it brings the genetic changes, DNA strand breakages and all. Okay, कौन करेगा वो physical करो या chemical करो, we don't mind. Okay, but still today I will tell you chemical cannot induce a directed mutation, or similar way the physical also cannot induce a directed mutation. Okay, so both mutagens will bring the DNA changes. That is the fact. Okay, this is one. Second is what? BRC, yes, we have a radiation facility. Because of that, we are using the radiation. Like you suppose if you don't have a radiation, you can make use of chemical, no issue.
Okay.
Only thing is, but the situation, circumstances are there where suppose you have a genotype, very rigid genotype. Even whatever you do it, you are not getting the change. Let us assume through chemical mutation. Then you can make use of higher doses of radiation to bring the gross chromosomal aberrations. This is where the radiation has an edge over chemical. This is one aspect. Then the second aspect is in particularly vegetatively propagated crops where you don't have a seed, you have a stems, you have a corms, you have a bulbs. Their size is very huge, where the chemical penetration in those seed material is very less. Whereas in for radiation, the penetration in these seed material is very, very good. So if they penetrate, they bring the breakages, DNA strand, this thing. And then the third aspect is particularly in the interspecific crosses. Okay, radiations will do bring the chromosomal translocations. In that way, they are advantages. But if you have want to have some point mutations, small rectifications, okay, you have very good variety, small thing you want to change it, then the chemical mutations are better. And then there are varieties from Dharwad, they are of EMS origin. Okay, so we have both radiation as well as chemical origin.
Okay, sir. Sir, you have mentioned radiation doses are also vary from genotype to genotype.
Yeah.
So it is very difficult to optimize the dose for a genotype because variability is very high.
No,
Is
sir.
any optimum dose for that we can apply?
No, that is where, see, in my, based on my experience, I will tell you in groundnut, okay, 150, 200, 250 gray can give you better results, no doubt in that. So
Yeah.
these can be blindly one can apply. But still, in my own case, I could find 150 gray could give nothing in genotype like TG37A, whereas the same 150 gray could give very good results in TAC24. Okay, I have both the extremes in my case. So, but still, I say that 150, 200, 250 will be better. But still, no, we don't know which genotype is having the specificity or the response to the radiation. That part we don't know. So, because of that, if you standardize, well and good. Otherwise, what? These 150, 200, 250, still, if you want to go for higher, 300 gray can be used.
Okay, sir. One more question, sir. Disease, very good example in disease resistance in mutation breeding, you have also mentioned and identify genotypes also. But it is possible for soil-borne diseases also. You have tried
No,
and
we have
obtained.
not tried. Even you remember, sir, Ratnakumar sir also had one stem rot
Yes, yes.
related mutation breeding project. There also he could not succeed. And then, sir, particularly these collar rot, sclerosium, and all, they are very, very tough fungus. Having resistance to them is very, very difficult. The getting the mutation for them is almost, I should say, impossible. But for that, we have very good wild species reservoir. The way from there we can make use of, initially bring those genes into our, and then we can improve our genotypes having that resistant gene. This is one possibility. Then the other possibility I could see in the last week in our institutional presentation from somebody, they could induce sclerosium related resistance also in some crop. Okay, there are examples, but I should say it is very, very difficult.
Because sir, resistance level is low, so it create resistance. It's very difficult. But what possibility can be can through mutation breeding?
No,
It
resistance,
is a
sir.
hit and try method. Hmm?
One can try, sir. One can try. For that, no, what we need is a very good sick plot screening methods. Okay. So if we have a very good sick plot and then continuously if we try, we can get it. Like in banana, wilt resistance they have obtained. Fusarium wilt.
How, sir?
Huh. Through radiation they have.
Radiation.
Not in our country, some other country. Okay, Brazil or Argentina. Somebody was presenting in our one of the IAE meeting. They were telling that they could get a fusarium wilt resistance in banana.
So they irradiate susceptible genotype or sir somewhat tolerant genotype?
I don't know exactly the in detail. In detail, we don't know.
So sir, mutants following Mendelian inheritance for traits.
Yeah. We have lot of studies. And then in our own हमारा बसु साहब का जो ग्राउंडनट बुक जो पब्लिश किया था उसमें हमारा एक चैप्टर है ओनली बीआरसी म्यूटेंट्स का ही इनहेरिटेंस का हमने दिया है मोस्ट ऑफ देम दे फॉलोड both monohybrid and dihybrid but as I showed you sometimes we do get some unusual inheritance patterns. Okay, because problem is with the groundnut. Groundnut is a polyploid, okay, autotetraploid, where that dose compensation, all those problems comes into picture. But still, um, we do get the mostly the recessive monohybrid ratios.
Okay, sir. Sir, these all the bark varieties are indirect mutant variety. Means mutants are used for the breeding program for developing the variety. That's it.
Correct, sir. Correct.
Yes.
See, as I tell you, no, TG1, TG3, TG38, TG73. Okay, these are the only direct mutants. Rest all are the mutant variety, mutant derivative. Okay, like that.
So it
But
means
but 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 in the pedigree, somewhere some mutant genome is there.
it, it means we have used other existing mutants for developing varieties or for high
Yeah,
yield
yeah,
other traits.
yeah.
Better to go directly as a mutant because it's a hit and try method.
Because
It
sometimes
will
what
be get
happens,
or not get.
yeah. Because sometimes what happens, you do get mutation for your desired trait, but in other traits, it may be lacking. And then ultimately variety means high yield. High yield is a must.
Yes.
Okay, in addition to yield, you need to have the other traits also. Okay, for that one should go for the recombination breeding.
Okay, thank you, sir, very much for my clarification. Thank you.
Veena sir.
Sorry, sir.
No,
Actually,
please, please.
sir, your presentation is very nice, and always we like to listen your whatever you will share your knowledge. But today it was very amazing experience. Sir,
Thank you, sir.
actually, I wanted to ask same question because in Rajasthan we are getting the problem of that collar rot. Because especially we are growing Virginia group and we are getting the polar rot problem in whole Rajasthan. So farmer are using 10 to 20% extra seed to maintain the plant population. But is it is not the way, sir. Actually, I wanted to develop a variety which can tolerate or resistant to that collar rot. So by mutation breeding, it is very tough. You already explained that one. So, sir,
But
which way we should try,
no,
sir?
still my suggestion will be, sir.
Yes, sir.
My suggestion will be like this. Okay, one thing is you have anyway the sick plots are there or the highly affected plots are there. Okay, what
Okay,
you can think of by having a little bit higher dose, irradiate your RG lines or whichever the parental, and then grow in that. Okay, and then also still you can inoculate physically also, and then identify some of the survival ones, and then again have a progeny testing in that. This is how you may get it. I am still doubting, but you may get it. Still, what
sir.
my another suggestion will be DGR, Rudachalam, and maybe other, wherever some of the interspecific derivatives are there, no, try to
Okay, sir.
screen them at your location, then find out some registered source. Those registered source in turn you can bring into your background through crossing followed by radiation induced mutagenesis, means to facilitate chromosomal translocations. That is a little bit bypass type. Okay, but that I feel will be a better idea than that first strategy.
Yes, sir. Definitely, I will try accordingly. And sir, for mutation breeding, first time I'm going to start the work at my center. If you'll see for protein, high protein content, high oil content. Large date we have many genotype for large date, but for high protein and for high oil uh, we are going to start the work by mutation breeding. So I wanted to take help of your center only because we do not have that mutation eradication facility and all that one, sir.
No, I really appreciate your idea, sir. But both the characters, protein and the oil, are quantitative in nature. One,
Okay.
okay. Even my PhD problem was on oil content only for mutation. Okay,
Okay,
but I
sir.
will share my experience.
Okay,
See,
sir.
first of all, you have to have very large population. One. The second is being a quantitative in nature, you have to have a screening method, particularly non-destructive. Like if you have NIR or NMR facility, the best, no doubt. Because each and every plant, M3 seeds, you have to screen to the extent of 20 to 30,000 plants. Okay, there you may get a variability of to the extent of you cannot get a big jump from 45 to 55. No. Okay, you can get what some 45 to 48 or 45 to 50, at most 52. Okay, instead of this, our Mishra sir का मैं formula बताता हूँ. Okay, instead of looking for 3% increase in the oil, you increase the 9% in the yield.
Yes, sir.
Okay, that formula looks better instead of directly targeting through mutation breeding. But still, sir, kindly try. Only thing is you need to have NMR or NIR facility to screen very large population. I did for my this thing from for tag 24 from 48, I could reach up to 52 only, even after screening through NMR. But only again, no, sir, this oil, no, 52 may show you at Durgapura. Okay, it may not be the case at Bikaner or maybe at Jodhpur or some other place. This is a highly variable trait. So, but when you have already very good high oil genotypes are available, better to take them rather than having going for induced mutation.
Okay, sir. Actually, sir, this time we have I have our one variety identified, which is having 48% oil content and 30% protein. It is, I
Okay.
think, sir, breakthrough for that butter industries.
Hmm.
What they are peanut butter industries. And again, I'm trying for same, sir, because it will be like a biofortification in groundnut.
Really, sir, really, really,
Yeah.
really.
And sir,
Again, sir, parallelly, you also see the amino acid composition of those proteins. Okay.
okay, sir. Okay, sir.
Because 30% is really on higher side. I think this data must be over the location and over the seasons, as I understand.
Yes, sir. For the for all season and for all locations, sir. It is
Fine,
nearly
fine.
30%.
Really,
Continuous.
sir. Really great, sir. Really.
Okay. Thank you, sir. Thank you so
Yes,
much for your nice presentation and nice explanation. Thank you, sir.
thank you, sir. Thank you, sir.
Ajay sir, Ajay sir, Ajay sir, Anandpur.
Hello. Hello. Good evening, sir.
Harish sir.
From Hiriur. Sir, myself Harish from
Hiriur,
Hiriur, sir.
Please, sir, please.
Congratulations for a very nice presentation, sir, and a wonderful
Thank you,
work.
sir. Thank you.
And I have a small one query, sir. Just now you
Please,
spoke about oil content, sir. Locations and seasons, there is a lot of variation. I think we also observed this one with respect to oleic acid content, sir. Oleic acid, sir.
Folic acid,
High oleic percentage. There is gene to E interaction is there with respect to this high oleic content also, similar to that of your oil content. So, are there similar observations made from your side?
Yes, sir.
high oleic?
Particularly oil content, I agree with you. They are really disturbed, particularly stressed conditions. The oil content will go down. If you have very good environment, very good management, definitely you will appreciate the oil. Okay.
Yes.
But if the stress is there, then the oil will come down. No doubt in that. Okay. So the same genotype, for example, in my location, it will give 48. The same genotype will give up to 52 in Gujarat. Okay,
Yes, sir.
this much variation is there because of the stress and unstressed condition for oil point of view. But oleic acid point of view, they are already well known to recessive genes are there.
Yes.
Wherever they are there, okay,
Yes.
around 75 to 80% oleic acid is ensured. Okay.
Yes, sir.
Only thing is they won't be much affected oleic acid point of view, whether you grow in the one environment stressed or unstressed, to the extent of some 5% here and there. But
Yes, sir. That
if you have
5% we have observed, sir. That's
yeah,
why I'm asking.
yeah. But if you have both the genes in recessive condition, my knowledge will be you do get high oleic acid of that 75 plus or so.
Yes, sir. 75 to 82 we have observed the range.
Yeah. That much will vary, sir. That much will vary because the seed also has to develop, no? So
Yes.
it needs unstressed condition.
Okay. Thank you, sir.
Oh, thank you.
No, sir.
Yeah, please.
Sir,
Ajay.
yes, sir, it was a nice lecture, sir. So we are
Okay.
hearing after a long time about this mutation breeding.
Yeah.
Sir, I have one small doubt. When we are going for a mutation breeding, in which generation it is better to identify those mutants, and then how long it takes for the mutants to get stabilized?
The best stage is M2. Okay. But if it is of maternal origin, means cotyledon, seed coat type of things, M3. Okay. So there you identify the mutant and then go henceforth, means in subsequent generation, plant to row progenies, continuously stabilize it. But still, uh, not only in groundnut, even in other crops, stabilization also takes time. Okay, sometimes still that segregation will continue. There are examples segregation continued till M8 generations also. So always whenever you identify mutant, let us say I identified in M2, go for plant to row. Again in that row, even all the plants, if they are showing the mutant trait, still then you identify a harvest individual plant, again you go for plant to row. Like that, minimum M6 generation, you advance plant to row only. After that, you try to bulk it, because what happens, not only that mutant gene, there will be lot of interactions are happening with this mutant gene with the background genome. Okay, so that will disturb when you go for bulking and all, stabilizing, attaining, as I say, homozygosity and homogeneousness. Okay, so that is a difficult. So best thing is advance it up to M6, plant to row. Then once you establish M6 onwards, you try to bulk it. So, but usually in our condition, we do after M10 only, we start considering that as a mutant and evaluation.
Because in some crops I have seen even after mutant characters they start appearing after M5 or M6, not before that.
Not only, sir, in tag 24 itself, अभी तो 30 years. Let us assume 2 generations per year means 60 generation, means M60 plus. I should say, still it is segregating for some traits. Okay. And then we have in sunflower, one genotype is there for seed color. Till today it segregates. Okay. So it is very, very difficult. Only thing is, in most of the traits, we could stabilize, no doubt in that. But certain traits are do segregate. But most of the economical traits, they get stabilized. But as I say, M6 onwards till M8 or so, that will be the ideal period to say a mutant. But every time try to go with plant to row.
Thank you, sir. Sir, I have one question.
Please, sir, please.
Regarding the QTL for Bruchid resistance, what is the progress for post that? Is almost 10 years gone. So any progress in that?
No. What happened? I will tell you the background. VG9514 from Rudachalam. Okay, again, that itself was an interspecific derivative from Arachis cardinacea. That line we could get it from Rudachalam. Using TAC 24 and VG95114, we could have an real population. Okay, mapping population. In that, of course, TAC 24 and VG9514 having the differences for the Bruchid resistance. When we subjected that mapping population to the bruchid infestation, we could get some of the QTLs for the resistance. Of course, VG9514 has a tolerance or resistance for the bruchid resistance. And then once after that, no, that real population and all, we left it, and then QTLs are identified, and then that much only. But there are a lot of things are there. Only we need a very good screening method, particularly in the interspecific derivatives. No? We will definitely get the resistance, no doubt in that.
Thank you, sir. Thank you. Bera, sir.
Yes, thank you. I think though number of participants are less, 42, but good interaction. So since it is a classical breeding, so most of the people interacted and maybe within their domain. So all agri people are mostly from classical breeding side. So I thank you, Dr. Anand, for good interaction and a good, very basic topic and lecture. I have two submission, not question, not any pointing out. Only two submission for idea of the participants. One, we used to say M2 generation, we have a large population. We must have when we are following the mutation breeding. What is that large? Can we quantify for groundnut for the knowledge of all the participants?
Yes, sir. In my opinion, sir, I have done in our own using tag 24, we could have 20,000 plus M2 plants. 20,000 plus M2 plants. Okay, sir. But still, because of the other reasons, in other genotype, I could not get I could not grow such a large population in other. Let us say I could grow some 6,000-7,000 only. There the frequency of the mutations were less compared to the one when I used 20,000 plus population. Okay. So in that case, the larger, but in groundnut, I should say 15 to 20,000 plants will be ideal.
So if we want 15,000 or 20,000 M2 plants, so it will come from M1. What would be the number of kernels for initial irradiation?
Oh, no, let us take this way, sir. 20,000 seeds of M2. Let us assume at the moment 100% germination, sir.
Yes, yes.
Okay, and then 10 seed per plant.
Yes.
Okay, so M1 will be something like 2,000 plants.
2,000 kernels, plants.
2,000. Okay,
And
so
again, we have to increase if there is a 70% or 80%, 90%.
So like that, usually what, sir, if we want to take 3 doses, for example, so better to have something like 1,000 seeds per dose.
Yeah.
Okay, and then that too for 3 doses will be ideal.
3,000.
3,000 seeds, taking care of these number of pods per plant, seed per plant, and the germination.
So
Will be ideal.
Ideal will be around 1,000 seeds.
Seeds per dose. Per dose.
Yes.
So for 3 doses.
Okay. So this is for clarification of the house. Another one thing, you have said from M2 onwards, plan to progeny, and if we practice that up to F6 or F7, again continuously we are increasing the number of plants. Suppose if we M2, if we select at least 100 plants or maybe 50 plants, and every plant will have as a constant figure 10 kernel, 10 pods, 20 kernels. So up to F6, what would be the population? Or we have to throw in every generation?
Yeah. Sir, very simple, sir. Let us assume 20,000 M2 plants. From that, let us say some 100 plants are selected. Now you will be having 100 M3 progenies. Okay, sir. In that 100 M3 progenies, all may not breed true. Let us assume 50%. So in that you have only 50 progenies. Within the, let us say 50 progenies, all they are true breeding. Okay, sir. In that per progeny, you select 2 to 3 best plants. Best plants.
Yes, that is the game changing. 2 to 3 best plants per progeny.
per progeny, sir. Okay. And then also in that one plant, you keep it a backup. Another one or two plants, you again raise it as a plant to row progeny in the M4. So from 50, again you will be having two plants, means again 100 progeny rows. If it is only one plant, 50 progeny rows. Not all the plants per progeny, sir.
Any progeny, if nothing is there, they can
Yeah.
leave it.
Simply reject it, sir. Simply reject.
Okay. Okay. So now more or less it's clear. Another one, that is as a director, how most of the breeders from the ACRIP center or DGR can access your mutation facilities, irradiation facilities.
Sir, anytime anybody can send the seed any amount, absolutely no issue to me. Okay, I will get it irradiated. Okay, sir. That too free of cost from my side.
That I want to listen free of
No,
cost.
absolutely. No, absolutely no issue, sir. Sir, not only, sir, even so many of our colleagues, not only in groundnut, even in other crops, like from Gujarat, castor, and then from others, sorghum, okay, PDKB and all from sorghum. Every year we do get some 50 to 100 seed irradiation request to us. And then we do, sir, no problem.
So we have listened Dr. Anand. So he is very kindful to help you. Those who are weak in hybridization or not able to generate your own generation, you are mostly depending on others. You may opt for this mutation breeding, and it is very simple as Dr. Anand shared here. You can send simply your parent material seed. He will be happy to irradiate and you can take forward. So this is one message. Second is, out of this today's topic, since we all are here, I want to just take your 5 minutes to discuss another burning issues. This is entirely different from your lecture because yourself is there and Dr. Shrikant is there from Chhattisgarh. I am here and Dr. Nataraja is there also. As you all know, Government of India has introduced that oilseed mission from this summer onwards, Rabi summer onwards. And in brief, I am telling they have prepared one document, that secretary. They have engaged one additional secretary, lady additional secretary to lead this National Oilseed Mission under the Secretary, Ministry of Agriculture, Government of India. So once it is rolled out and there is a clear-cut plan, they have based on the prospect of the each oilseed crop in each states, they have identified district-wise particular oilseed crop and cluster demonstration. Any district of any state is good for groundnut, cluster demonstration of groundnut will be there. Similarly, for all the mustard, soybean, then sunflower, they have designated and it is a big document. So similar way, they have allotted cluster to Assam and Chhattisgarh. And Assam, there is no variety released by state government nor CBRC. Similarly, Chhattisgarh, no CBRC variety, only two variety. One is from that Raigad center and that two bark variety and one bark line and one from ICAR-IIGR line. And they asked us to give the list of 5 years old varieties for different state groundnut. They want to supply seed for that cluster, and state will go for a cluster demonstration all over India, 10 lakh hectare per season. So Assam, it is a very difficult situation. We told there is no variety released for Assam, neither through state, neither CBRC, but we can try for any variety which has been recommended for Zone 4. Then immediately they told, no, your some Zone 4 recommended variety, you have mentioned only Odisha and Bengal. Very obvious that 2 locations we have tested. So that's why council has mentioned it. We have mentioned it, but we can go for it. But they are telling, if you suggest, we can recommend it. Secretary is telling, if something happens, then it will be risky for us. Minister or parliament will say again, you have not followed the right procedure. So it will be a problem for ministry as well as your directorate as well as ICR. Will not go for that. So ultimately, ADG seed from ICR, he suggested we can go for a 1-year trial, agronomy trial, which we take on after during AVT stage, so that we can take the any popular varieties or any varieties recommended for that zone but not recommended in that state. And once we test it and get a result, after that we can go for a area expansion. That is fine. Where I want to raise the one pinpoint, Raigor scientist is here. He is here, Srikant. So he has released 2 varieties from the state, and when we asked the breeder seed quantity available with him, they are planning, ministry is planning for seed supply in this Rabi summer, as well as they are planning from next Kharif, next Rabi and next summer, what would be the breeder seed available from the breeder. And all the 3 next season, Srikant mentioned 0, 0, 0, nil, nil, nil for 25-26. So if this is the situation, if we are not able to produce the breeder seed, may not be this Rabi summer, Kharif 2025, we are not able to give anything. Rabi 2025, matlab next Rabi and summer 2025-26, we are not able to give a 1 quintal, 2 quintal breeder seed. Then what is the use, or I should say fun of releasing such type of varieties? This is my submission, not blaming, not fingering out anybody. So if we are releasing variety, please multiply your breeder seed as fast as possible and help me to, from this such type of sarcastic comments from the ICR as well as ministry. And you
Sir,
all know, when we sit for with the meeting with these bureaucrats people, they simply blame us. They are not in a position to listen our words. Simply say there is no variety, or this institute has not released any variety for that particular state in last 10 years, last 15 years, like that. What they are doing.
yes, no, sir, I want to add here, sir. the Chhattisgarh point of view. Okay. In the Rabi season, we could multiply some 3 quintal plus. And then based on that, the National Seed Corporation, maybe from the Delhi or somebody, they told them. So last 15 days or so, sir, they lifted 3 quintals of that TG88, that is CGTM, and then took it to the kernel for the multiplication, and then shortly it is reaching Kurnool in Andhra Pradesh, sir. Okay, sir. And then in addition to that, already I have shown a program for the production of 20 quintals during this summer 25 already, sir. Sowing is done, sir, already. Okay, so I am expecting, sir, around 20 quintals or so of this variety in the summer itself, means by May end or so. Okay. So this
So
is one part.
why this information Dr. Srikant is not providing, being a nodal breeder of that varieties? Srikant
Srikant?
is a nodal variety of that your variety as well as that ICAR-IIGR variety, Chhattisgarh Mungfali 1 or something like that.
Sir.
Simply, Nataraja is sitting here, has mentioned 000, and secretary is telling next 3 season, Chhattisgarh will not have a single kg of breeder seed of that release varieties. What Dr. Jadhav proposed, and Chhattisgarh asked to the government, central government, you give the seed of Kadri Lepaksi. And Kadri Lepaksi is not recommended for Chhattisgarh. So I have been given the task, you go for trial of Kadri Lepaksi 1 year in Chhattisgarh and recommend. This is the fruit I got because Dr. Srikant has reported 000 for all the next season breeder seed availability.
No,
He
sir,
has released 3 varieties, and this is the paper he has submitted, and I have to pay the penalty for that.
No, sir. I am in touch with him always. And then I have indicated him also that we are multiplying the seed, of course, in a smaller quantity. And then before reporting, just a phone call away. Okay, sir. If you would have, before writing rather, okay, he should have consulted. I would have given my number, sir. Absolutely no issue. But anyway, sir, but this summer also we are multiplying, sir, from BRC point of view. And then if nature favor us, definitely we can get some 20 quintal plus. And then definitely, sir, whatever possible from our side, sir, we will multiply and then supply. So our motto is to give the seed of these new varieties, whoever is possible. And then, sir, in addition to that, this tag 73, in addition to PDKV Akola, of course, Akola is doing in a big way. In addition to that, another some 30 quintals or so I supplied during this Rabi season, and then another 50 quintal plus program is already sown, sir, in this summer 25. We will be expecting some 50 plus quintal for that tag 73 also, sir. In addition to Maharashtra, it is doing well in Madhya Pradesh and Andhra Pradesh. Wherever you feel, sir, we can give this seed of this variety, no issue, sir. Then, sir, Assam, you were telling, no? Just for your information, sir, what happened? Of course, Assam don't have any variety, and but in recently when they were evaluating some of our material, TG37A and TG51 till today, based on their conditions, they found it better. If that is the case, sir, I don't know intricacies or the procedure point of view. Based on this result, if they re-recognize or re-notify, or I don't know what all those procedure, if that is the case, immediately, because they have some data of their own local MLT data, if they do it and then re-recognize or whatever, straight away, sir, 37A or TG51 centrally sponsored varieties are there. We can push in the Assam region, sir.
No, they have put a mark on only 5 years variety.
Yes,
Only
sir.
5 years
Okay.
old variety. They are promoting through this oilseed mission. So they have put that rider.
Rider is there, sir.
Yes.
Mm-hmm.
So definitely before this workshop review meeting, we are going to take some decision to divert the release of such variety by borrowing the material from others and not producing breeder seed again. For those centers, they are just simply releasing through state and not providing seed. Again, there will be a big penalty for them, those who are doing and not taking breeder seed, foundation seed production, all those things. Because they are putting me in trouble. And Nataraja is here. If I am telling wrong, anybody can verify from Dr. Nataraja. He was there also in meeting. So that is my concern. So that's why I have raised. It is totally.
Yes, sir.
Yes,
sir. We are multiplying. No, sir. As I said, no, around 20 quintals or so in the summer, we are multiplying, sir. Whether the IGKV multiplies or not, we don't mind, sir. But we are multiplying.
Why IGKV is not multiplying? They have.
I cannot comment on that, sir. I
They have
cannot
released.
comment on that.
They have released. They could have taken the nucleus seed from you or consent from you, and it is their primary responsibility.
Yeah. I fully agree with you, sir.
He is the nodal breeder, and their university has released as a state variety. It is the primary responsibility goes to that breeder.
Yeah. I agree, sir. I agree.
How long will continue with spoon feeding from ICAR-IIGR and bark? Why to allow for releasing of such variety if we are not able to produce seed? So, okay, thank you, Nataraja. You can conclude.
1 minute, can I want to give one.
Yes, sir, please.
Yes, sir.
Okay. Actually, Anand, thank you very much for this educative lecture. And
you, sir.
Please give preference for boldness and early or medium duration, and also for this high oleic lines with early duration, like in Spanish blood. What about your this one, our advanced lines with high oleic lines on par with or in with Girnar 4 and Girnar 5 in yield and other things?
No, sir. Now we have developed, sir. Now that large scale evaluation in line with the Girnar and all are to be evaluated. Now we have developed, we have confirmed. Now they are in F12 or so. Now after evaluation, sir, we will push in the system, sir.
Okay. Okay, because
then,
Girnar 4, Girnar 5 and GG40.
But as you are telling the bold seed, no, it is only Gujarat appreciate the bold seed, sir. Elsewhere, lot of marketing issues are there. Okay, the procurement and all is a big problem. Followed by also the equally good management, agronomy is also equally needed. And then if something goes wrong in the management, no, the seed size comes down, shelling comes down. That is a big problem, sir.
To see that how it is far better than Girnar 4, Girnar 5 and GG40.
Sure, sir. Sure.
Okay. Because it is the future of groundnut, na?
Sure, sir.
Okay, thank you. Thank you, Bharat.
Yes, sir. Thank you.
Well done, Anand.
Dr. Anand, you can unshare your PPT. Yeah.
Yeah.
Somebody was telling something. Anybody? Anybody want to interact? Or any
Somebody.
question? Somebody.
This somebody was me, Dr. Bera.
Oh, sir,
Arey
sir, please open your video, sir.
No, I don't know. I'm using my mobile.
Okay.
Okay, fine. Your voice is fine. Your
Good
voice
evening,
is
sir.
enough, sir.
Good evening.
Good evening, sir.
I wanted to hear Anand, so I didn't take rest this afternoon.
Sorry, sir. Sorry for disturbing you.
No, I enjoyed.
Thank you, sir. Thank you, sir. Thank you, sir.
Well done.
Thank you, sir. Thank you, sir. Thank you, sir.
Sir, apologies. I could not attend your. I missed your lecture, sir. Some urgent work.
There was nothing special in that. You know what I say all the time.
But it is always pleasure to listen you, sir.
Well, there would be other occasions. Don't worry.
Yes, sir. Yes, sir. Yes, sir.
But this is a good initiative that you have taken.
Thank you, sir. Thank
And
you,
I
sir.
hope these young breeders, they take advantage of this.
Sir, one is I am taking very colloquial language. One side is GAN. GAN, what will take, what will use, what will deliver, that is a separate issue.
Yeah.
First thing, we must interact, join, come together, discuss, interact.
That is true.
That gives a very, very impactful environment.
No, that is very much required, and actually it should not end up with these lecture series only,
No.
These breeders should interact with each other. They should also interact with senior breeders and I'm sure everybody is willing to help and guide.
Yes.
So
when they are facing something in field or lab or writing paper also, they can discuss with senior anybody.
That's true. Let's hope it makes some difference, even if 5 to 10% difference is there. I think you have achieved your goal.
Thank you, sir. Thank you. And also, I appreciate all the seniors' participation and their kind help and their labor also, because all have prepared a good PPT and they took their time to prepare the lecture, PPT, and for presentation. Sir, heads off, sir. I really acknowledge your support and system, particularly from DGR and all ICAR-IIGR. I bow my head to all my seniors and experts, those who are cooperating and helping us. Thank you, sir.
Right. Thank you, sir. Thank you. Keep doing good work.
Thank you, sir. Nigam, sir, thank you.
Okay, Professor.
Nigam, sir.
Yes.
Yeah, Anand here, sir. Sir, kindly, based on our today's this thing, kindly guide us also. Okay, in mutation breeding or combination of that, what are the lacunas, kindly point out so that we can also rectify now through mutation breeding.
Right.
No, sir, Nigam, sir.
Ji.
For that, Anand has to provide that Sandha Puja.
Oh, of course, that is.
Sir, Udaipur is already destination is fixed.
No, I'll try to come there, Udaipur.
Yes, sir. We'll have a good Sandha Puja.
Right, Jarur, it is important.
Yes, sir.
Right, sir. Thank you, everybody.
Thank you, sir.
Right, sir. Bye.
Thank you, sir. Thank you, Anand, sir, for the good lecture and interaction. I hope all the participants have gained some inputs, insights into mutation breeding. And thank you for extending the olive branch that you will be helping any ACRP science centers or groundnut breeders with mutation breeding exposure to irradiation. Thank you, sir, for that. And sir, if you are okay, then we'll put this Zoom recording on our YouTube channel, IIGR YouTube channel.
Yeah.
Thank
Yeah,
you, sir.
definitely, sir. You can share it and then you can give it to anything. And then also, sir, not only groundnut, even we are all extending even in the other crop. So many scientists, they do take up the mutation breeding, particularly in the vegetables where it is lacking. Not only in India, whenever we attend the International Atomic Energy meetings, even the Central Europeans, the Asian countries, and also they are taking our guidance and all. Again, next week, again, I am going. Another 15 plus countries are there. Then there also, I am really proud, sir. They all appreciate our program being done, and then success happened at BRC in groundnut, and then the support what all we are getting through both DGR and our all SAU partners, then success of these varieties. Always at the international fora, they really appreciate us. So I'm really thankful, sir, particularly personally giving me the opportunity to director, sir, so that I can share my experience, sir. Thank you very much, sir.
Thank you. And
Thank you, sir. Thank
Dr. Nataraja, you can announce the next lecture date, time, and
Yes,
speaker.
sir. So next lecture is scheduled for 10th of February. So Monday at same time 2:30 to 4. And that speaker will be Dr. R.D. Prasad from IIOR, Hyderabad. So he has developed some formulations, seed treatment formulations. So he will give that lecture, sir. So I've already shared the Zoom link details. In that topic has been mentioned. So please join that meeting, sir.
And requesting all the participants to encourage the students, particularly MSc and PhD students, from working under you or under your departments. Here at DGR also, Doctor, please invite.
Yes, sir. We are inviting, sir. Today, 6 or 7 students have joined, sir.
Yes, yes.
Yes, sir. From Udaipur also several people have joined, PBC students.
Thank you.
Yes, sir. So, sir, now we have one brief presentation from ARS Kadri, Dr. Venkatesh, sir. He was leading the team